going to be sharing stuff that, in some cases, me and my collaborators have been working on for several years, and in some cases has really just appeared in the last few weeks. And so it's something broadly that I'm calling neurovascular modulation. And these are my disclosures, and my slides are online, or you can scan this link. I'll put it up at the end also. So I'm going to be talking about a mechanism for that spans all forms of neuromodulation: deep brain stimulation, invasive stimulation, magnetic stimulation, ECT, tDCS, and so on. And I'm not proposing a new form of neuromodulation. I'm proposing a new way to think about all these forms of neuromodulation, whether they be based at a center or based at home. And they center around the fact that these stimulation modalities may actually be affecting the brain vasculature. And I think the major clue for that is something that we all know very well already, is that if you image the brain's hemodynamic response, you see it. And so that's sort of the clue that starts the whole process. So I don't need to tell this audience about neurovascular coupling. I do want to draw your attention to this sort of the capillary density that you can sort of see in that video. So the distance, which really surprises me, between these capillaries on average is about 50 microns. And so I think that's really important when you think about neuronal arbor, right? Or a dendritic arbor. It's really, you're talking about neuron capillary. It's unbelievably dense. And everyone knows that when the neurons are active, their metabolism increases, and they can call for more vascular activity in order to support it. And that's sort of that neurovascular coupling. And then, though this is a research topic, the notion is that increased vascular coupling will then enhance and support neuronal metabolism. So it is a two-way street. And again, as this audience knows very well, this is the basis for many forms of hemodynamic-based imaging, whether it's something like fMRI or something like fNIRS. And this has been deployed broadly to study the effects of neuromodulation. So the general idea is that neuromodulation will stimulate neurons, right? That's what we've been hearing about, and that's what I've thought about for many decades. Those neurons, by being active, will then trigger a sort of dynamic hemodynamic response. And then we have very fancy tools to measure that response. And then from that response, we will infer what happened to the neurons. But the point is that these hemodynamic signals are considered epiphenomenon, right? They're not actually interesting themselves. It's kind of like a means to an end. That's what I mean by epiphenomenon. And so this is not an original cartoon, but I couldn't find the original, so I've redrawn it myself. And so so the suggestion here, of course, is, this child is getting a gift, and they're just so thrilled by the wrapper. They're not even looking inside. And so I'm suggesting that maybe something similar is happening with the richness of data that we have using hemodynamic imaging to look at brain stimulation, that there's actually something deeper sitting there. And so what I'm suggesting is that we can have a situation with the neuromodulation, the application of energy into the brain, whether it's via TMS or electrical or DBS, is actually directly activating the brain vasculature, whether it's large vessels or small capillaries. And of course, then we can image it using hemodynamic imaging, and then we are measuring that vascular response. Now, if you stimulate the brain's vascular response, you will affect neurons as well. But it is reversing the order of typical neurovascular coupling, where we sort of think of the neuron as the master and the vascular as the slave. Now it's vascular first. And I think this reversal of recruitment order, as I'll discuss, opens up some very interesting ideas about therapeutic mechanisms, but it also obviously opens up some questions about mechanism to begin with. I need to say up front here, there is no doubt that the neuromodulation is directly stimulating neurons. That's something I've worked on myself. We've heard talks about it. I've worked with brain slices where they have no vasculature. So I'm proposing this as a parallel or alternative mechanism, but I'm dangling hemodynamic imaging here because there's something tantalizing about it. And so again, it's still talking about neurovascular coupling as a two-way street, but it's asking, could neuromodulation directly activate the vasculature directly? And this is a little bit hard to untangle because of neurovascular coupling. And it's very hard to stimulate one without seeing a response in the other. And our default will always be to assume that we've stimulated the neurons directly. And so I'm going to be focusing initially on whether this question may be unpacked for transcranial direct current stimulation. It's something I've been interested in for a long time. tDCS, non-invasive application of a battery type current across the head. And one way to ask the question is, well, all right, we have the neurons, they're in the parenchyma, you have glia, and then we have the blood vessels here. If the neurons were not there, would the system still respond, right? So you take, and then that would sort of let you address this question in part. Obviously, that can't be done in vivo, but it can be done in vitro. So there are reduced preparations, essentially endothelial cells that form the blood-brain barrier that are being grown in a flat dish. And these endothelial cells attach themselves to one another. So you effectively form a barrier. So you can imagine like a long two-dimensional sheet of endothelial cells that are stuck together, and that sheet from top to bottom will form a barrier. Now, there's a lot about this that does not look like the blood-brain barrier, but this is a system that has been proven to have utility in asking certain questions about the blood-brain barrier. So this is what that system looks like. These are the endothelial cells. They're grown in this dish. The dish has a mesh on the bottom of it. You can see they're forming this tight barrier. There's fluid over the top and the bottom. You could think of the fluid at the top as the inside of the blood vessels, the inside of the lumen, and you can think of the fluid on the bottom as the space outside where the neurons are. And so that's your blood-brain barrier going from top to bottom. And these systems have been studied for a long time. And you can ask questions like, you can look at transport across the blood-brain barrier. You can look at different size molecules and how quickly they move across the blood-brain barrier. But you can also, and this is important for what I'm going to talk about, you can look at water transport. So there is a pressure gradient from top to bottom, but the endothelial cells resist that flow, and you can track that resistance using that bubble. So it's literally a bubble, and the rate that bubble moves tells you the rate of water flux through the blood-brain barrier. And so there is a low and controlled level of water flow from the inside of the blood vessels, from the lumen into the extracellular space, into the parenchyma. And that is a feature of the blood-brain barrier, and it is a feature that can be captured here as well, as long as assessing the transport of different solutes that can be fluorescently labeled. And also you can, afterward, look at different structural functional effects on these cells. The only difference we've done, this preparation has been around for decades. We've just added electrodes. So we stuck a wire in the top chamber. We stuck a wire in the bottom chamber. We connected that to an electrical stimulator. And so now we can zap these endothelial cells using different waveforms, direct current, TMS relevant wave currents, and so on. So now it's an established preparation. We're just generating a known level of electrical stimulation across them to see what happens. And we've been doing this now for a few years, and this is the single most, for me, important result. It's one of those results that is nice because it's trial by trial and you can see it by eye. You know what I mean? So it's those kind of effects. What you're seeing here is the movement of that bubble, which shows you how much water is moving across this blood-brain barrier model system. And you can see that there's a general velocity of this bubble, and that velocity, the displacement is shown on the left, and the velocity is being plotted on the right. You see there's an initial drop in velocity. This has to do with this model system where the cells seal together tighter as the flow begins, but then it stabilizes. And then we turn on the stimulation. Now with direct current, we can do positive terminal on top or negative terminal on top. So we have two possible directions. So positive terminal is anode, negative terminal is cathode. And what you can see is that when we turn on the stimulation, the bubble moves at a different rate. And depending on which way we turn on the stimulation, we can make the bubble move faster or we can slow it down. What that means is that we're pushing water. So we can push water. This effect is very reproducible. It's intensity dependent. And it is, in this case, acutely reversible. And like everything else, it's, when you think about things and you look at the literature, it's one of those things that should have been completely obvious. There's a phenomenon called electroosmosis, where under certain conditions, electrical current will drag water with it. Certain conditions need to be in place in order to maximize this effect. Electroosmosis works best when there is a tight channel and where the surface of that channel is charged. Now, this effect is not happening across the cells, it's happening between them. So even though these cells form a barrier, that barrier is not perfect, and there's a little gap between them, which they call a tight junction, because a tight junction. But that's where a lot of the transport happens. When we apply electrical stimulation, it is easier for the current to travel between the cells than to go through them. Some will still go across them and polarize them, but if you have a gap, that current will find the gap. As that current travels through the gap, it's traveling through an extremely small channel, and that channel has negative charges on its surface. That's the outside of the cells. So this is the condition under which you get electroosmosis. The amount of water flow that we see here is precisely predicted by electroosmosis once we consider the features of these endothelial cells. So, and it should be intensity dependent. It should persist as long as the current is applied, and it should reverse. So this phenomenon is well captured by electroosmosis. And so now we're seeing something, though. We're seeing a coupling between electrical flow and fluid flow. And that's the key, right? We've gone from electrical now to mechanical. Now, it turns out that when you drive flow through these endothelial cells, that is a very powerful signal for the blood-brain barrier, meaning just changing flow in itself will trigger all these secondary responses in these endothelial cells. They will transduce not the electrical flow, but the fluid flow, and a whole bunch of secondary things happen, and these can be quantified. And again, I'll refer you to the publications, but a lot of interesting things happen. First of all, molecules get dragged along with it. How this is just because you're producing fluid flow, and so the molecules will, at a basic level, will be dragged along with it, even if you don't actually change the structure of the barrier. How much they will be dragged along will depend on their size, and you'll see that larger molecules have a larger relative change. And we also see a bunch of structure-function relationships. Again, I think this is because this is a direct consequence of the fact that we produce the flow. I don't think this is actually electrical stimulation of the endothelial cells, so that could happen. I think these are things that are actually very well expected to happen as a result of flow. And I think these changes can also include activation of early genes. It could activation of molecules that would be active both on the blood-brain barrier, but also possibly on the neighboring neurons. And so it's not, so from electroosmosis, there is this cascade, and it's complicated cascade of effects. I like to use the term plasticity here because these things can now outlast the stimulation. The electroosmosis will not outlast stimulation, but these other things can. So when we love to talk about plasticity, and now I'm saying I'm using that term here very intentionally to ask, to think about plasticity of the brain vasculature or plasticity driven by the brain vasculature. Now, I've shown you data for direct current. We've done experiments using biphasic high-intensity pulse current that might be roughly analogous to maybe DBS or SES or a particularly long train of TMS. And we see effects as well on, here you're looking again at the movement of this bubble, this water transport. Now, in this case, you see that 5 minutes of stimulation produces a change in water transport that outlasts stimulation. And we've also looked at the cellular and molecular consequences of stimulation at different waveforms, and you see different effects. There's a lot to unpack, but the bottom line is that the way the vasculature responds to stimulation depends on the waveform, and that's fine. That means that we have waveform-specific effects, which we already know from neuromodulation. I'm just saying now that those waveform-specific effects could potentially be explained in part by how they differentially affect the vasculature, though they all affect the vasculature. In this case, we saw just 5 minutes of stimulation, producing a profound increase. And again, we can look at where those lasting changes may come from. In this case, for example, staining for specific proteins that are involved in the formation of the tight junctions. And again, I'll refer you to several papers where we've looked at this as well. But it is, for me, not a question anymore whether endothelial cells in a dish will respond to electrical stimulation. So I think that answers the question, at least in a very reduced system, do these cells respond? And the answer is they can respond. So what does this mean? Why is this potentially interesting? And so I'm calling this neurovascular modulation, just as, instead of neuromodulation, to emphasize the word of the vascular involved. I think it's interesting in many ways. first of all, just the ability to boost brain metabolic capacity is interesting. There's many situations where we would like to do that, perhaps in the context of neurorehabilitation, where we're trying to train the brain, and we know that plasticity is going to be metabolism dependent and metabolism modulated. So we can think of almost any example where you're looking to boost brain function, and that's a lot of neuromodulation in itself. And I should mention we've extended this work from in vitro to in vivo models. These are models with mice where we can maintain the skull, shape away part of it, and then use multiphoton imaging to visualize individual capillaries and the space around them. Now we have neurons and capillaries present, so it's limited in that sense, but we've extended these findings to that system as well. But another area I think which is very interesting is just talking about the water transport. So now we have a way to basically literally flick a switch and change water transport across the brain. We drive water across the blood-brain barrier, and therefore we're driving water across the interstitial space. This is the space around the neurons. And so, it can use sort of the colloquial term flushing to say, well, now can we flush the brain? And for those who study different types of diseases that may be associated with abnormalities or plaques in the extracellular space, this sort of rings some bells. Of course, this is very speculative, but I want to talk a little bit about that, and it's something that we've looked at in different ways. So there's kind of 4 steps to this process that I want to unpack. The first one is that when you apply electrical current through the brain, that current weaves and winds itself around neurons, and also weaves and winds its way around capillaries, and some of that current will cross into the blood-brain barrier. The blood-brain barrier is unbelievably resistive by design. It's like when I have to find the value. It's like strangely high resistivity value. And so a tiny amount of current produces a huge electric field, and I'll show you how much. But we think that you get about a 100, sometimes 400x amplification. That means a 1 volt per meter field around neurons can produce a 200 volt per meter field in the blood-brain barrier. A 100 volt per meter field, this might be produced by TMS, right? Now we're talking about thousands of volts per meter across the blood-brain barrier. And I'm going to talk about that because that conjecture is controversial. We know that any electric field across the blood-brain barrier will drive water flow. We've also separately shown that when you apply direct current in vivo, the transport of solutes in the interstitial space, so this is now not across the blood-brain barrier, but between the neurons, also increases, right? So there's direct evidence that not only does water flow across the blood-brain barrier, but this has consequences for solute transport across the interstitial space. And finally, I'm going to show you some models that try to scale all this up to make specific predictions. So the first point about this concentration one, and I think this is interesting because I've gotten a lot of criticism of this. And so usually with modeling, when you get people angry, you at least know you're onto something. You may be going down a dead end, but at least it's interesting. So we basically said that we think there's this amplification. So we did this multiscale model, first predicting the electric field, in sort of a voxel of brain, and then projecting a capillary network into it. So you have to imagine that this capillary network has a highly resistive wall, and then the inside of it is actually rather conductive because it's blood, and the outside of it is actually relatively conductive as well, and so the current is flowing through it, and it doesn't really want to cross the blood-brain barrier, but when it does, it hits the blood, and now it's like another avenue for it, and so you have obviously different factors at play that models help you consider. And what we found is that there's this huge amplification. So in the case of tDCS, usually 1 milliamp will produce a fraction of a volt per meter, but across capillaries, we're predicting 160 volt per meter. And we showed in this paper that we call neurocapillary modulation that in theory, this would apply to any form of neuromodulation. And this is where we get to these 10,000 volts. So this is kilovolt per meter, right? We're saying that TMS, ECT, DBS are going to produce kilovolt per meter across the blood-brain barrier, and people really did not like this for many reasons. Though obviously the models are the models, and it's interesting. So I could say a lot more about this, but there's a lot. Well, the question people ask, first of all, that seems too high, right? It seems like that's like electroporation level kind of stuff, and we haven't seen any damage. And they also think it's a bit unclear because usually when we talk about polarization, we talk about polarization across the cell membrane. And here I'm not talking about polarization across the cell membrane. I'm talking about polarization of like a barrier, which includes a bunch of endothelial cells. And so people really wanted some unpacking of this. You know what I mean? And I thought, okay, that's very fair that we're being a bit vague about what we mean about these kilovolt per meter fields. But anyways, I refer you to this paper for a lot of the theory. And so this took 2-3 years to develop these new models. These are multi-scale in the sense that we're going from centimeters, like the whole head, down to nanometers, which is the tight junctions. And these models have to be run in stages. And so you have to make certain assumptions across the scales, obviously, that the details of the one lower scale will not affect the higher scale. Even so, there was a lot of things we needed to consider. The head models are well known. The capillary models, we expanded from what we did before, and we had to consider multiple types of capillary networks and how they might be connected into larger networks. And then we had to go into the level of the blood-brain barrier. So now you're looking at a cross-section of a vessel, and now you're getting down to nanometer scale. So these are these tight junctions. They're extremely narrow little channels, and that's where we believe the current is concentrating. And I'll refer you to the paper. There's a lot that had to happen, but the central notion is that you predict the electric field in a voxel, and from the voxel scale, you predict the electric fields at the micrometer capillary scale, and from there, you need to predict the electric fields at the nanometer scale inside the tight junctions. Inside the tight junctions, electroosmosis happens. So now electric fields become fluid flow. But now we got to scale the fluid flow from the nanometer scale up to the capillary scale and then back to the whole brain scale. And so there were a lot of conceptual steps here, but the notion is we're trying to go from a neuromodulation dose to a prediction of how much do we change fluid flow at the macroscopic level. And what's interesting is once we went through this whole pipeline, it turned out that in many levels, there was a lot of linearity, such that at the end of the day, we said, well, subject to assumptions, it's very easy to go from electric field to fluid flow. It's basically a constant. And that constant is dependent on many, many, many things, but it's one constant. And so, for example, here you're seeing simulations of two tDCS montages, the M1SO and the bifrontal. The bifrontal is used for depression. M1SO would be used for different motor disorders. We can stimulate at 1 milliamp or 2 milliamp. And in each case, we get a current density prediction. These are pretty conventional. But what you're also seeing is for each case, a brain-wide fluid flow prediction. And this is a linear multiplication of the regional electric field. And so we're saying that when you apply 1 milliamp of bifrontal tDCS, this is how much additional interstitial fluid flow you will produce. So it's per minute, but it's like milliliter. It'd be like a volume of fluid flow per minute per volume of brain. So it comes out as a per minute thing, but we're basically saying you have a voxel of brain. How quickly are you exchanging the fluid flow in that voxel of brain? And the values that we were predicting here are comparable to endogenous rates of exchange, which means that when you apply tDCS, you can double the rate of interstitial fluid exchange. Now, this is just a prediction, and there's a lot of aspects to unpack with it. But it's consistent with the notion that you could use something like any neuromodulation technique, including tDCS, in order to flush the brain. Now you could say, well, okay, will it actually work for things like dementia and so on? That I don't need to convince you of, because those trials are already ongoing for other reasons, right? So I don't need to convince you to run the trials. The point here is to suggest that there's another mechanism that may be in play other than the one that was always considered, which could be fluid flow exchange, but it may also be just general capillary stimulations. And many neurological disorders are associated with capillary dysfunction, right? And so now you're treating the capillaries. You're not treating the neurons directly. So boosting brain function, driving brain clearance. And finally, one more idea, which has to do with using stimulation as a neuroprotective role. This is not a new idea, but I'm just putting a suggestion that maybe now neuromodulation can help protect neurons, facilitate a neurorehabilitation by directly stimulating the vasculature. I've done a lot of work with this with Dr. Bar Hosseini, who's a neurologist at UCLA. The animal literature was already there. And recently, Dr. Barhosani designed a trial in which these were patients, these were acute strokes, and acute, I mean, they're being rushed into the hospital. This is an emergency situation. Within 10, 15 minutes, she and her team were applying stimulation to these patients. This is image-guided stimulation, where every person has an identified specific injury, stroke, and the stimulation is being applied in a way that is customized to that individual, and they were assessing outcomes. This was just recently published in JAMA Network Open. It was a pilot trial, but the results were remarkable. And I'm just listing this in an example where now, inspired by the fact that you can directly stimulate vasculature, new trials can be designed, and I believe these are ongoing as well. So those are the ideas that I wanted to share. I know it's really quick, but the references are there, and thank you for your attention.